Okay, so back in the 1920s, we understood that chromosomes had the genetic material. The question then became with what component of a chromosome carried the genetic code, whether it was the DNA or the proteins. They knew the chromosomes were made up of both of them. So DNA was a likely culprit because DNA, they knew, was very diverse. It was made up of 20 different amino acids in various combinations, and so therefore had lots and lots of different functions. So it seemed kind of likely that that would be something that could be diverse enough to hold the genetic code of organisms. The other idea was that DNA was the molecule that held the genetic code. And the evidence for that was that the DNA was found in the right place, it was found in the nucleus, that it was in the right concentrations or the right amounts. In other words, if they looked at the body cells of organisms, it had a certain amount of DNA. When they looked at the egg or the sperm cells of the same organism, they had half the amount of DNA. And then the last was that the DNA in different organisms was found in different amounts, but when they looked within the same species, each member of that species had about the same amount of DNA. So that's kind of where they were, and so we're going to go over some of the experiments that were used in order to determine that DNA was in fact the genetic material. So the first experiment that was done by Griffith. And what he did was he was working with bacteria, and he had this one strain of bacteria. Let me see if I can get a pointer that works better than the one that I have. Okay. He found that he had this strain of bacteria, and he called it the smooth strain, or the S strain, because the outside, when you look at it under a microscope, appeared to be smooth. This particular strain had a capsule on it. And there was another strain that lacked this capsule, and under the microscope it didn't have a smooth surface, and so he called that the R strain, or the rough strain. And when he took the S strain and injected it into a healthy mouse, the mouse died. And then if we follow Koch's postulates in determining what causes disease, we have to then isolate the original organism, the original pathogen, from the organism. So then what he did was he did a necropsy, or an autopsy, on an animal and looked at the heart, and he found the S strain cells. So that was in fact what was killing the mouse. And then he did the same thing with the R strain. And when he injected a healthy mouse with the R strain, the mouse was healthy, and when he did a necropsy, he did not find the bacteria cells in the heart. Then what he did was he took the S strain and he heated it to damage the cells. And so he damaged the cells, and then he took those damaged cells, injected a healthy mouse, and the mouse stayed healthy, and when necropsy was not able to isolate these cells from the heart. So then what he did was he took the R strain, the strain that did not cause disease, and he mixed it with the heat-killed S strain, which also in it itself did not cause disease. And when he did that and injected it into a healthy mouse, the mouse died, and when he did a necropsy, he found R strain healthy cells growing in the heart of the organism. So what he realized from this was that something was transforming these cells. So these cells that were not virulent, did not cause disease, were somehow transformed, these cells were somehow transformed by these cells that had been damaged. And so he called it a transforming factor. So they were looking to figure out what this transforming factor was. So the next experiment actually showed that DNA was the transforming factor. So in this case, what happened was Avery took these heat-killed bacteria cells, and he grew them up, and then he filtered, and he homogenized them, like blended them together, and then he strained them. So then he had just the components of the cells. So then he took a sample of that, 3 samples of that, and in the first sample, he used an enzyme that destroys RNA. That's the RNase. And in the second sample, he used protease, which is an enzyme that destroys proteins. And then finally, in the third sample, he used DNase, which was an enzyme that destroys DNA. And then he grew these up with some of the R strain, just like before. So he took this material, mixed it in with the R strain, just like Griffith did. And when he did this, the first two he was able to isolate the S strain from it. In the one with the DNase, he was not able to get the S strain, R strain only, which meant that this prevented the transformation. These two tubes did not prevent the transformation, so RNA was not the transforming factor. Proteins were not the transforming factor. However, DNA, when that was destroyed, transformation did not occur, so it was the transforming factor. Another experiment furthered supported that DNA was in fact the genetic material. And this was done by a couple of scientists, some last name Hershey and Chase, so it's known as the Hershey-Chase experiment. And they used bacteriophages, viruses, and infecting bacteria. So before we talk about their experiment, I want to talk a little bit about a virus and how that virus infects a cell so that you can understand their experiment. Okay, so this is a bacteriophage, and let's see if I can get my marker. I noticed on the other one that the little arrow, you couldn't see it. So here's my bacteriophage, and if you look at a bacteriophage, it actually has a really elegant structure to it. This is a micrograph of it up here. So here, it has this protein coat, and inside of the head, this protein coat is the genetic material, and in this case, it's DNA. It also has this structure here which kind of can act like a spring, and then it has these little spiky leg kind of things. Now viruses have no metabolism, so they have no way to generate energy or do anything. However, if this virus attaches to the surface of the cell, and it's specific to this particular type of cell, this is a bacteriophage. We call the viruses that infect bacteria bacteriophages, sometimes just phage for short, and this one's named T2. So what this does is it attaches to the surface of the cell, and once it's attached, it's a more stable conformation if it kind of squishes down here. And so as it squishes down, it injects the DNA into the cell like a syringe. Now the outside of it stays on the surface, just the genetic material goes into the cell. And then that genetic material takes over the cell, makes the cell become basically a viral factory and produce lots and lots and lots of viruses. Eventually it bursts the cell open, and all of these viruses are released, and they can go on and infect more cells. So what they did was they labeled these bacteriophages with different isotopes. And so they used sulfur and they used phosphorus. And let's go to the next slide. So we know that DNA contains phosphorus. Remember, it's sugar, phosphate along the backbone. And we know that some of the amino acids that make up proteins contain sulfur, such as methionine and cysteine, and we saw that earlier in the year. So they used a different isotope of the phosphorus, and they grew up the viruses. I don't want to call it growing up, but they allowed the viruses to reproduce and be made with only this particular phosphate present. They did the same thing with the sulfur. So they had these two separate labeled bacteriophages. They then put them into a solution with bacteria, allowed them to infect the bacteria. They homogenized it again so that now any of the parts that are on the surface of the cell would get removed, and whatever was left inside of the cell would remain within the cell parts. And then they centrifuged it. They spun it down so that the heavier components went to the bottom and the lighter components went to the top. So in this case, the cellular components made a pellet at the bottom, and the fluid that it's around made up the supernatant. And when they did this, what they found was that the labeled phosphorus was in the pellet, or that was in the cellular component, and that the labeled sulfur was in the supernatant, meaning that it did not make its way into the cell. So that further supported the fact that DNA was that transforming factor, that DNA is the genetic material. So once they determined that DNA was in fact the genetic material, the next thing came to be is what is the structure of DNA? So another scientist working in the 50s, Chargaff, did different studies of different species, and no matter what species he looked at, he was comparing the different nucleotides. He was comparing the different bases of the DNA. He found that there was always about the same amount of A as there was T, or this is adenine, and this is thymine, and that the amount of C or cytosine was always equivalent to the amount of guanine. And he called these purines and pyrimidines. So the A's and the G's are purines. Oops, I didn't mean to go forward. And the T's and the C's are pyrimidines. So one way to memorize this, because it is on your essential knowledge or demonstrate understanding, is that A's and G's are purines, and you can think of angels and God are pure. And then CT is Connecticut. Those are the other two, and I don't know, Connecticut doesn't have any pyramids. Pyrimidines, pyramids, I don't know. But that's one way to think of it. The other thing is that it's always going to be a purine with a pyrimidine. So A's go with T's and C's go with G's. And one way to remember that is that A's have straight lines and T's have straight lines. So those go together. And G's and C's are curvy, and so they go together. So they knew that there was this equivalent amount of A's and T's and C's and G's. And then there was a woman working in King's College in England, and she was doing X-ray crystallography. And so what she was doing was she was working with another scientist called Maurice Wilkins, and they were taking DNA samples, and they were using X-rays to a beam of this X-rays would go through the sample, and then it would create this diffraction pattern, and they would develop that. And so they would end up with some picture that looks like this. Now, I look at that, I see a bunch of dots, looks pretty. But other people could look at this and say that this is representing a double helix. Now, I can't see that by looking at this, but there was a woman who taught AP Biology for a long time. Her name was Kim Foglia, and the way she explained to her students what this picture showed was as if you were trying to figure out the structure of a crystal chandelier without actually looking at the chandelier. So if you think about a chandelier hanging with all these little crystals hanging off of it, and if you were to take a light source above the chandelier and shine it down on the chandelier, and all you could look at is the shadow pattern on the table, and use that shadow pattern to predict the location of the crystals in the chandelier. That helps me a little bit get my head around it. I don't know if it helps you, but anyways, that was her contribution. And then James Watson and Francis Crick continued. These guys were working at Cambridge in England. And they were the ones that actually finally came up with the structure of DNA. I shared with you the article that they published in Nature magazine. But this is their model that they built out of, I think they used tin. Anyways, they were the ones that are credited with finally determining the structure. They were awarded the Nobel Prize along with Maurice Wilkins. Rosalind Franklin did not get the Nobel Prize because she passed away, and they don't award the prize posthumously. Okay, so they came up with the structure of DNA, and I'm not going to get into a lot of detail here, but we know that it is a double-stranded helix. We know that the diameter of the molecule throughout the entire molecule is uniform. It's right-handed. I have a hard time getting my head around that, but that has to do with the way that the molecule is. It's not like a perfectly symmetrical helix. You can kind of see that it sort of twists different, right? These kind of bands are staying together, and then you end up with this larger opening, and so on. It's anti-parallel, meaning that the strands run in opposite directions, and that finally these nitrogenous bases are exposed with this major groove here, and so you have this larger opening here, and then this minor groove. Let me get my little pen, maybe that'll help. So a major groove here, and then a minor groove here. Okay, and we also know that there's the complementary base pairing, and we know that the bases are found in the middle of the molecule. So it goes sugar, phosphate on the outside, and the bases are found on the inside. And we know that A's go to T's and C's go to G's. We also know that there are 2 hydrogen bonds between A and T, and there are 3 hydrogen bonds between C and G. And that is it for the structure of DNA. The next lecture will be on DNA replication. Thanks for joining me.